Today we will go into the subtopic which focus on the protein synthesis. Basically, the protein synthesis consists of these two major processes. Okay, one will be the transcription. Okay, in which this focus on the copying of genetic information of the DNA. Okay, and this will produce the mRNA. If you remember, when we talk about the central dogma, we should know that the DNA has got a connection with the production of proteins. Okay, so this will be the first part in which the genetic information contained in the DNA will be copied through the process of transcription, and this will be producing the mRNA, and this occurs inside the nucleus. Okay, and the second part will be translation, that occurs in the cytoplasm, in which the genetic information that is now within the mRNA will be translated to produce polypeptide chain, or the proteins the proteins. And these two processes of protein synthesis will be consisting of several stages. Okay, and so for the transcription, there will be these three stages. And number one will be initiation, number two elongation, and number three termination. Okay, the same goes for translation, which can also be divided into three stages, also named as initiation, elongation, and termination. However, the events that are occurring during these stages will be different from the initiation, elongation, and termination for transcription. And another different thing is that the elongation stage in translation will involve three steps, okay, that will occur repeatedly. Okay, the steps will be codon recognition, peptide bond formation, and translocation. Okay, we will look at the stages after this. But before that, before we focus on the protein synthesis, okay, let's look at this relationship between the genetic code and the sequence of amino acids. Okay, this also has got something to do with the central dogma, in which we should know that the DNA, which contains the genetic information, okay, in the form of a sequence of the nitrogenous bases, okay, can be copied. Can be copied through the process of transcription, and this will be producing the mRNA. Okay, so we should remember that the DNA molecule consists of two DNA strands. Okay, as you can see, this is one strand. Okay, and this is another one. Okay, and in the process of transcription, only one of the two strands will be copied. Okay, and that will be known as the DNA template strand. Okay, meanwhile, the other strand is considered as the non-template strand. Okay, and so based on the DNA template strand, okay, which is also known as the antisense DNA strand, will be copied, and from that template, this will be produced, the mRNA strand. Okay, and usually we will read the sequence of the organic bases in the multiple of threes. The triplets of bases of the DNA will be copied, and from there these codons will be produced. Okay, what are codons? Codons will be the triplet of bases of the mRNA. These codons will later be translated, okay, translated through the process of translation, in which one particular codon will code for a particular type of amino acid. Okay, for example, the codon of AUG will code for the amino acid methionine, and the codon of AGC will code for the amino acid serine. Okay, GAC for aspartic acid, CAC for the histidine, and so on. So there are many types of amino acids, and all will be coded by the codons of the mRNA. Okay, and so through the process of translation, all these amino acids will be joined together to form a long polypeptide chain, also known as the proteins. This is a genetic code table in which we can see that the combination of three types of nitrogenous bases, okay, can create a particular type of codon, and this will code for a particular type of amino acid. Okay, for example, okay, if the first letter is U, uracil, the second letter is also uracil, and the third letter is also uracil, U. So the codon will be UUU, and this codon of UUU will code for this amino acid known as the phenylalanine. Okay, and so there are many types of amino acids that can be coded by all these different types of codons. Don't worry, you don't need to remember all, but you need to remember four types of codons. Okay, codon number 1 that you need to remember will be AUG. Okay, this AUG codon is special because this is the codon that is also known as the start codon. The start codon will be the codon that will tell us where the process of translation should start. This start codon also code for a particular type of amino acid, and that will be the amino acid methionine. So you just need to remember AUG, the start codon, and it codes for the methionine. Okay, there will be 3 more that you need to remember. Okay, and this will be the codons of UAA, codons UAG, and codon UGA. All these 3 codons will be special because they are known as the stop codons. The stop codons, in which these stop codons will tell us that the translation should stop at that particular point. We will continue now with the process of protein synthesis, and we will start with the first type, that will be transcription. Okay, in which I've told you before this that transcription is the process in which the genetic information of the DNA will be copied to form the mRNA, and this occurs in the nucleus. And this transcription will be showing 3 stages. Okay, the first one is initiation, in which in the DNA there will be this promoter site, that will be the first part of the gene. Okay, and there will be a particular enzyme that is important, that will be the RNA polymerase. Okay, so in the process of transcription, there is one enzyme that you really need to remember. The enzyme will be this one, the RNA polymerase enzyme. So in the process of initiation, the RNA polymerase will just try to attach to this promoter on the DNA. Okay, and then we will continue with elongation, in which we can see this red thing over here will be referring to the RNA strand, and the RNA will be produced, and it will become longer and longer. So the elongation will be basically referring to the elongation of the RNA strand or the mRNA. And of course, this will be ended by the process of termination, in which there will be one part of the DNA that will be known as the terminator site. Okay, so when the DNA polymerase enzyme over here reaches the terminator site, okay, the process of transcription will end. Okay, as you can see in this particular animation. Okay, now let's look at the stages in detail. Okay, we will start again with the stage number 1, initiation. So what is important for you to remember about the stage 1, initiation? Okay, you just need to remember that there will be one important enzyme in this process, that will be the RNA polymerase. Okay, and there will be this DNA molecule. Let's say this is one gene on the DNA molecule, and usually at the starting point of a gene, there will be a site known as the promoter. During initiation, the RNA polymerase will look for the promoter site on the DNA, and the RNA polymerase will attach to the promoter of the DNA. And once the RNA polymerase is attached to the promoter, the RNA polymerase will unwind the double helix structure of the DNA. Okay, as you can see, okay, once the RNA polymerase is attached already on the promoter, it will start to unwind the double helix of the DNA. Okay, and there will be two, of course. There will be two strands of DNA, but only one will become the template to produce the mRNA. So which one? Okay, normally there will be one strand that is known as the antisense strand. Okay, that will be chosen to become the template to produce the mRNA strand after this. So during this stage number 2, the RNA polymerase enzyme will simply move, okay, or slide along the template strand of the DNA. And as the RNA polymerase enzyme move along the template, it will catalyze the addition of free RNA nucleotides. The free RNA nucleotides will be joined together to form this RNA transcript, also known as the mRNA. Okay, and as the RNA polymerase move along the template, the RNA transcript will become longer and longer. That's why we say that this stage is known as elongation. Okay, and the elongation of the mRNA will occur in the direction of 5' to 3'. This will be continued with stage number 3, the termination, in which this will happen once the RNA polymerase reaches a special sequence on the template that will be known as the terminator site. Okay, the terminator site. So maybe somewhere here at this particular template strand, there will be this terminator sequence, and once the RNA polymerase reaches this terminator sequence, the RNA polymerase will be released from the DNA template, and the mRNA will also be released from the template. Okay, and now this mRNA or the RNA transcript will be having codons, okay, that will later be translated during the process of translation.